Hi everyone, welcome back. This is me. I'm still here in my little office and I am doing my best to use this computer to talk about root uh, function, specifically how roots uptake nutrients in the form of ionic substances from the soil. So we're going to go through four things: the absorption of ions by root hairs into the epidermis, the movement of the water and ions through or around the cells of the cortex, and then the movement past what we call the Casparian strip and into the vascular cylinder where it meets the xylem and starts being able to be transported by transpiration. And then there's a really cool video at the end about mycorrhizal fungi if I have time. Otherwise, I'll just post it on Moodle. And I apologize in advance for this computer going crazy. All right, so one of the major root functions, obviously, is moving mineral solutes in the forms of cations and anions from the soil that the plant is hanging out in into the plant itself. So it turns out that the charge of the ion makes a big difference in how it behaves and how the plant needs to move those ions into the root. So I'll just remind you from intro chemistry that anions are negatively charged ions and cations are positively charged ions. The stupid way I remember this is that anions starts with AN. Anions are negative ions, and that cations are positive. Get it? Because like cats have paws. Yeah, you've probably heard this before, but it is a useful memory tool. All right, so anions that are important to plants include, I'm going to point out nitrate, phosphorus, and sulfur. All of these are really important. Chlorine as well, but nitrate is the main one I'm focused on here. It turns out in solution that the anions are easily available, relatively easily available to plants. It doesn't cost energy to move anions into plant cells. However, because they are freely available in the soil, the anions are easily washed away if there's a flood or if the soil is overly irrigated. So there's a trade-off there. Cations, so positive ions that are important for plants, include calcium, which is important for proton pumps, magnesium, which is important for chlorophyll. Remember that chlorophyll has a magnesium ion in the center of it, so that's obviously important to get up to the leaves. Potassium and ammonium are also very important for building proteins like rubisco and other important plant molecules. So cations, on the other hand, compared to anions, tend to be associated with clay particles in the soil. So there's a picture of that here. You see these are brown blobs are clay particles, and they are associated here because clay tends to be negatively charged as a sort of a mixture of substances. It tends to have a net negative charge. It tends to sort of attract cations in the soil that sort of stick to it and make those cations difficult to get into the plant. So there's that problem. And the second problem is that moving that cations into the roots is an energy requiring, ATP requiring process. So plants basically have a particular issue getting cations into the soil. Anions have their own problems being washed away. And so we're going to talk about some of this in a little more detail. Okay, so it turns out that the cell wall itself is not a barrier to ionic movement into the plant. So the cell wall, made of mostly cellulose, is permeable to ions. On the other hand, the plasma membrane tends to exclude hydrophilic materials like ions because the plasma membrane is primarily made of lipids. So ions can't just pass as they wish across the plasma membrane into the cytoplasm of the cell. So for both cations and anions that are necessary for the plant's nutrition, the proteins that are embedded in the plasma membrane have to assist in the uptake of both kinds of ions. And so we're going to be talking about protein pumps and transport proteins very briefly, not in that much not in that much detail, but we'll do the best we can. Okay, that was my alarm saying that I'm supposed to be in office hours in 10 minutes, so we'll do my best. Okay, so the transport into the root, as I was mentioning, involves proteins involved in the membrane, and I'm going to very briefly just mention the three ways that proteins are important here. The first is a proton pump. So proton pumps are establishing an electrochemical gradient that allows cations to move through the cell along the gradient. So let me explain that a little bit. The proton pumps lead to a strong excess. So the proton pumps are pumping hydrogen ions outside the membrane of the cell, so the space between the cell membrane and the cell wall. And so there's going to be an electrochemical gradient, a proton gradient between the membrane and the cell wall. And that allows cations to move through the membrane along the gradient, because there is a net gradient between the outside and the inside, and these positive ions kind of by diffusion want to move into the cell. So this establishment of the gradient allows cations to move through the membrane inside cells that have root hairs into the epidermal cells. The thing that I want you to know about this is that the proton pumps are ATP requiring processes. So ATP is required to, remember ATP synthase, it's a similar kind of process. This kind of proton pump of moving protons outside the cell requires ATP. So moving cations into the cell is an energy requiring process. On the other hand, anions, negatively charged ions, actually have a different problem getting across the cell membrane. So because there is now a electrochemical gradient such that the cells are more have more protons or more positive ions outside the cell, negative ions can't move by diffusion through the cell membrane into the cell. They need co-transporters, and I'm not going to go into the details of how co-transport works. You can read about it in your intro textbook or in your plant bio textbook in chapter 30, but let's just say that there is a process by which anions the plant needs sort of move with protons across the gradient in channel proteins that are specifically designed, well, let's not say designed, that are adapted for this purpose. Okay, I'm going to move on. A little bit more about cation exchange, just to explain it one more time with slightly different pictures. Remember that cation exchange, or positive ions moving into the cell, requires some sort of removal of the cations from how they are attached to the soil particles. So the picture over here that I want you to see is that cations are attached to this clay soil particle because they are positively charged and the soil particles have a net negative charge. So in order to get those cations sort of released from the soil particle, the plant actively releases CO2 from the root hair, CO2 that is a product of respiration in the roots. So the cells are respiring, they're producing ATP, they release CO2 into the soil, and it binds with water and eventually actually allows hydrogen ions to be produced when carbonic acid is made. So plants encourage the exchange of protons that they are actively releasing from their root hairs. Their protons that were in the plant now are going to bind to the soil particles, which allows the cations to be released and moved into the root hairs via the active transport process that I talked about in the previous slide. So cation exchange refers to the sort of trading. These days I'm thinking a lot about barter. So the barter of protons that the plant releases that it's not needing, and releasing from the bonds between the cations and the soil particles, the cations that the plant actually needs. And so then they can be moved into the root hairs on the epidermal cells via active transport. Okay, so you should read more about that because I think what I said was slightly confusing, or come and ask me questions in office hours. Okay, so let's pretend that all that made total sense. And in these epidermal cells now, inside the cytoplasm, we have cations and anions and water, all of which are being absorbed through the root hairs via a variety of processes, either diffusion in the case of water, or transport through proteins in the membrane for anions and cations, with some ATP being spent. So now we have our substances, our solutes from the soil in these epidermal cells. Okay, that's great, but what we really need them to do is get those cations, ions, and water molecules all the way over here into the xylem, because it's the xylem that is going to move the substances into the rest of the plant for metabolism, photosynthesis, production of secondary chemicals, etc., etc., and growth, of course, too, from the meristems. So all of these substances need to move. Now, there's two ways that can happen. One is the sort of cheaty way. So the apoplastic route, you'll remember way back when we talked about cells, cell walls, we talked about the apoplast. It turns out that apoplast is the space between cells. So sometimes ions and water will move through the plant in the borders or boundaries in the cell wall, because remember, anions and cations can get through cell walls, but they can't get into the cell membrane without protons or proteins. So apoplastic movement, it occurs when the solutes are moving sort of around the cell membranes and into and sort of all the way over here to the pericycle, the endodermis, the border around the vascular cylinder. They're moving sort of only in the cell walls, and they're not entering the actual cell. Okay, so that's the apoplast. We're going to leave that there for a minute. The other way is the symplastic route. So the water moves in via diffusion, and then the solutes move in via the protein transport, as we were talking about. And then what happens is, once those solutes are in the epidermal cytoplasm, they can move through plasmodesmata, the channels between cells, and they're moving basically in the cytoplasm between cells all the way through from the epidermis into the cortex, which is primarily parenchyma tissue, all the way again to this epidermis. And then they move through the epidermal cells through channel proteins here that allow the plants into, or that allow the plants to move the water towards the xylem, which is these pink cells, these vessel elements here. Okay, so solutes can move through the symplast, and they actually arrive because they were filtered, so the proper amount of ions and the proper kind of ions were allowed and filtered by the root hairs, and then later by the endodermis. Those solutes are able to move through the plant easily. Apoplastic movement actually needs a filter, and the filter for apoplastically transported ions is here in the endodermis. So there is something I need to tell you about excluding ions and making sure that the plant only transports the ions that it needs into the vascular bundle, and not ions that it doesn't need that might harm the plant. So plants do need to keep some ions and some solutes out of the rest of the plant, like from entering the vascular cylinder. Plants have a sort of a security system, I metaphorically call it, and that security system is in this border of cells between the cortex and the pericycle inside the, so that it is bordering the vascular cylinder and the pericycle. And that the border cells are called the endodermis, and between endodermis cells, there is a sort of a seal. It's a substance called suberin, and it is impermeable to apoplastic movement. So apoplastic movement, for example, here, like here are some ions trying to get through the cell wall, and they're trying to move into the xylem, which is in here somewhere. And these ions are turned back because they meet the suberin of the cell walls. So those ions are excluded. The only ions that are allowed through are those that pass through channel proteins in the membrane of the endodermis cell. So the endodermis in the root has a really important function of filtering the solutes and only allowing in the solutes that are actually required by the plant at that time. So the endodermis membrane proteins are very important for moving solutes from the apoplastic movement into the actual cytoplasm of the plant and eventually into the xylem. There's some more pictures of this. Here's a picture of the Casparian strip, which is these cells here, which are sort of, you can sort of see it here. They have a sort of a seal. There's a block between these cells of the endodermis that's preventing movement of solutes out here moving through the cell wall. They hit the Casparian strip and they can't get in to the xylem, which is in this area here. Okay, sorry guys. All right, one more picture of this. This is sort of a cartoon. So this is an endodermis cell, and it has the Casparian strip, which is made of suberin, which is water impermeable, and it is incapable of allowing solutes through into the cytoplasm. So that the suberin is really important in the endodermis for filtering solutes from the environment that might have eluded the channel proteins in the root hairs. Okay, that's all I'm going to say about this. Nope, it's not. One more picture. Okay, here we go. So here is here's a picture of how transport works. So either symplastically, solutes enter through the cell membrane here and then continue to move through plasmodesmata like this, and then eventually into the xylem, or in apoplastic movement, the solutes move into the cell wall. The ions are able to get into the cell wall, but they can't get into the cell wall through the Casparian strip. They have to go into the cell membrane via channel proteins, which are not really pictured here, but that's what that arrow means. They're being moved either by active transport or by sort of co-transport through specific proteins that are only allow some ions in. So that's actually how some salt tolerance mechanisms work. Like salt can get into here, but the salt won't be transported into the xylem and starve the plant that way. All right, I want to switch right now and show you a really cool video about mycorrhizae, which I haven't basically mentioned. So I did mention that the root tips are in communication or sending signals and receiving signals from other organisms in the soil. Many times those organisms are mycorrhizal fungi, so fungi in the soil that aid in the uptake of solutes and other and water both, and in return the mycorrhizae actually receive carbon from the plant. So we mentioned that CO2 is exuded from the plant, but also polysaccharides are exuded from the plant, and the fungi actually are able to consume those. So this is what we call a it's not exactly symbiotic, but it's a mutualistic relationship between many different plants, especially forest trees, we're finding out, and the fungi in this, the fungi species in the soil. Okay, so I'm going to shut up and listen, and if I can get this to work, we're going to watch this. Oh my gosh, I am serious, you guys. I'm losing my mind here. Okay, I can't wait for my computer to come, but it's not here yet. Okay, let's see what we can do.
I want to take a closer look at these symbiotic relationships between plants and fungi. I've come to this ancient forest in South Wales to find out what goes on underneath our feet. Even just walking through this ancient forest, you can smell the soil. It's wonderfully fresh and earthy, and this is partly because of the abundance of microorganisms that it contains. I've come here to gather soil samples to analyse just how much fungi this ancient soil contains. The soil within this forest has been untouched by humans for centuries, and so it's developed a really amazing structure full of humus. This humus is only able to form when there's a mature web of soil organisms, including fungi. So I can't wait to get these samples back to the lab and analyse them. I'm not just looking for mushrooms, as these are only the fruiting body of the fungus. Instead, I'm looking for tiny thread-like structures known as hyphae. Hyphae are the thread-like structures that make up the fungal body, and together these strands make up a huge network known as mycelium. Underneath the microscope, you can see the hyphae in detail. You can see each cell that makes up the hyphal threads, and these all link together to form a pipeline for nutrients, enzymes, and fluid to pass through. Mycorrhizae are a type of fungi that form symbiotic relationships with plants. The plants capture energy from the sunlight and use it to convert carbon dioxide and water into sugar. The plant uses this sugar for cell growth, but up to 80% is returned back to the fungi in return for water and nutrients, which the fungi gets from the surrounding soil. Fungi and plants have been cooperating together since plants first colonised the land 450 million years ago, and since then fungi have formed relationships with up to 95% of plant species. In untouched ecosystems, fungi are intrinsically linked with plants, but within many soils worked by man, these mycorrhizal fungi sometimes struggle to thrive. Farmers and gardeners alike can utilise the power of mycorrhizal fungi by incorporating them into the soil that they work. This pea seedling was only planted 14 days ago, and you can already see a huge difference in the size and vitality compared to that of this seedling, which was planted at the same time but with no mycorrhizal fungi. Incorporating mycorrhizal fungi into the soil and protecting it from further disturbance enables fungal populations to thrive. They are at the base of the soil food web, so once they're happy, the whole system can flourish.
Okay, so that's all I have here. I actually think mycorrhizae are probably the most important organisms on Earth. Don't tell the plants, because they allow plants to communicate with each other and to absorb water and solutes from the soil easily. It's really interesting, a huge growth area of research. So I hope you can read more about it. I just posted a couple, like a short popular article here, but there's an enormous number of scholarly articles about this too. But this is a plant biology class, so we're mostly focusing on individual plants. But mycorrhizae, watch this space. They're really cool. All right, that's all I've got, and I'm going to see you soon, I hope, and I hope you're all doing well. Take care.